right, good morning, everyone. The session 4 will be the tumor evolution and microenvironment. As you know, the neuroendocrine tumors are complex and diverse, characterized by different grades and differentiation, emphasizing the importance of understanding disease evolution, and also the importance of the tumor microenvironment in terms of in regulating cancer progression and therapeutic outcomes is now widely appreciated, highlighting the importance of further investigating neuroendocrine tumor microenvironment. So we will have first 2 talks about disease evolution this morning, followed by other 2 talks about tumor microenvironment by using single-cell transcriptomics.
Okay, so now I would like to welcome Dr. Etay Ziv. I hope I pronounced this correctly. He's an interventional radiologist at the Memorial Sloan Kettering Cancer Center, and today he will talk about one of my favorite subjects, which is the progress from low to high grade pancreatic NETs. Thank you.
Okay, thanks so much. Thanks for the opportunity to speak to you today about our work on tumor evolution. This is how I control this? Yeah. Okay, and I want to acknowledge the AACR and NETRF grant for their support and all of my collaborators. Himanshu is a postdoc in my lab, did the bioinformatics work. And of course, the center of all this is patients and families. I should say this is unpublished data. So the start of the story is really this paper from one of my colleagues, Laura Chang, a pathologist at MSK, who was studying pancreatic neuroendocrine tumors that had mixed grade, high grade, low grade regions at the same time. And what she showed was really these tumors still behaved clinically like well-differentiated neuroendocrine tumors. They were like not as aggressive as the poorly differentiated. Genetically too, they didn't have the p53RB loss. They had MEN1, DAX, and ATRX. And then in the discussion section of her paper, she kind of mused over 2 possible ways that these high-grade components arise, right? One would be sort of an accumulation of genetic alterations over time and it goes from low grade to high grade. And then the other way would be maybe that they were just distinct cancers in the same patient. And so when we started thinking about this, we knew that there were these mixed grade, but it was, I think the consensus was that it was sort of a rare thing, a rare event in a rare cancer. So we wanted to see how rare. So what I'm showing you here, each, the y-axis is Ki67, these are years. Each line is a patient, and I don't have a pointer, do I? Yeah, I do. Each line is a patient, and each circle is a time point where we're sampling that patient. You see that some patients stay low grade for many years. Some patients sort of slowly increase over time. There are some patients that spike up really quickly, and then some patients sort of even decrease over time. So if you tabulate all those, you have some patients, these ones that go all the way to high grade, it's 42%. Then de novo group D, those are the ones that Laura was sort of studying, that's 14%. And these ones that are sort of on their way maybe to transforming. So actually it turns out it's quite common, right? It's over 75% of these patients are transforming. And so, yeah, so transformation is common, probably the rule rather than the exception. It's fair to ask, why do we care? So of course, high grade really does drive prognosis in these patients. And so if it's this simple kind of idea of sort of genetic alterations over time, we thought maybe there would be some correlation between the number of mutations and grade, and it doesn't seem to be. So the relationship between high grade and low grade is not clear. There may be some question about mutations driving this. Laura showed that there were no p53 loss in her cohort, but really if we look at our cohort, really there's about 10% in the whole MSK cohort at least that have p53 mutations, and these are all truly well differentiated. And so maybe there's something driving that, the transformation. And then of course, as clinicians, there's some kind of anxiety, I think. Maybe some of our treatments may be promoting this transformation. Certainly alkylating agents cause hypermutation. Some people worry that that may be driving some of this. There's treatments we do that change the microenvironment that potentially is driving this. Okay, so with that in mind, we wanted to get a better sense of the relationship between these high-grade, low-grade components. So we took these 2 groups, the group A and the group D, Basically found specimens from different sites, different time points, and asked our pathology friends to help identify those and microdissect. We did whole exome sequencing at 250x. This turned out to be big bottlenecks because a lot of these tissues sort of failed these quality control steps. But what I'm going to show you now is 17 patients we've done, 10 in group A, 7 in group D, aligned the reads, got the varying allele frequencies, and then used triomics, which is one of these phylogeny inference algorithms out there to infer the phylogeny. So this is the first view of the phylogeny of a transformed PNET. Not this, in 1 second I'll show you, but just to kind of orient you, oh, there it is. Sorry, 1 second. So just to orient you, this patient had a primary tumor and a liver metastases that were resected at time point 0, a bunch of other treatments, and then at year 9 and 10, 2 more specimens taken. And so this is their phylogeny. Okay, so to start, I want you to look at this area. This is the primary, and these are metastases, and they were all resected at time point 0. They were all grade 1. And their relationship is what we'd expect, right? The primary is sort of upstream, and these metastases downstream, sort of like as we'd expect, sort of things evolving over time. And then 9 years later, they had another metastases that was sampled, and that turned out to be this high-grade region. And then another year later, an intermediate-grade region. And that was really striking to us, right? This was not at all what we expect. MET8, we'd think, would be somewhere down here, or at least somehow closer to the primary, but sort of maps to this early progenitor of the phylogeny. And you might ask, okay, maybe this is multiclonal. Maybe they're just different tumors, like the sort of second hypothesis that Laura had. But in fact, they have the same MEN1 truncal mutation. So these are the same, arise from the same progenitor. DAX seems to sort of give off or sort of align with each one of these separate branches, and appears to kind of arise early. And so coming back to sort of Laura's sort of initial hypothesis, it doesn't seem to be multiclonal, but it's not quite this neoplastic progression thing. It's sort of this really divergent early event happening, and then many different subclones, and some of those have then transformed to high grade. So how common do we see that pattern? And so I'm going to show you a bunch of more figures like this. So just to kind of orient you, these are years this is a single patient, years, and these are sort of treatments that the patient underwent. These are time points where the tumor was sort of sampled. And so you see, again, the primary tumor here, adrenal metastasis 5 years later here, a liver metastasis here, and that's the high grade. So again, early emergence of this high grade. Not at all what we'd expect. And we see it again and again over and over, this pattern, high grade mapping really early on the phylogeny. And so is that the only pattern we see? I'll just sort of make a quick point here. We'll come back to this ATRX DAX sort of occurring in the same place, but a little bit more explain about that. But that's not the only pattern. So there's other patterns we see where the high grade is rising where we'd expect downstream of in the phylogeny. And so sometimes we see this pattern again here. And so if you compare these, right, so what's the difference between these groups? So one simple thing, and maybe a trivial thing, is that these all went through surgical debulking. This patient had no surgery. This patient had surgery, but only the primary was resected. These other metastases weren't resected. So maybe there's a simple explanation here, right? So if you have surgery, you sort of remove all of these subclones, and then over time they can't transform. And then there's some other metastases, or somewhere else something's hiding, and then it eventually transforms. And that I think that is the explanation for this. I mean, certainly we have to do more, but there's also some deep things I think going on here which are kind of interesting. First, so as you know, neuroendocrines is one of the only cancers I can think of where if you remove 70% of the tumor, that works. That's a good thing. Like every other cancer, that's not at all a thing that you would do, right? And so this is one explanation of that, right? So if you have this early high diversity, this very branched phylogeny, and each of these subclones has some probability of transforming over time, the more subclones you remove, the fewer subclones there are to transform. And since high grade really drives the prognosis, if you can remove all those subclones, you're just turning back the clock and less likely for things to transform. And so that's maybe an explanation for why this surgical debulking works. The other interesting thing I think that's going on here is, all right, so it means that there's something dormant going on, right? There's some subclones hiding, right? These guys were resected as far as every part that the surgeon saw was removed, but then some of these subclones are hiding somewhere. They're dormant for years, and then at some point later they kind of emerge. So I'll switch quickly to these mixed grade, the sort of ones that were mixed from the beginning. The phylogenies look different. Won't go too far into it just from time. I'll just show you some interesting things here. Sometimes these are spatially right next to each other. So this high grade and low grade region are right next to each other. And they map to the same phylogeny. But sometimes they don't, right? So this is a high grade region going to here, and there's a low grade region that maps to this part of the phylogeny. They're both in the primary tumor. They sort of evolved somehow separately. It's interesting that this P53 sort of is sitting at the top of this branch. And you might ask, remember, Laura saw no P53 loss. We see 10% in the sort of full MSK cohort, but in our cohort, which are transformed, it's actually quite high. It's 44%. So is P53 driving this transformation? Certainly in some cases, maybe, right? This is a high-grade region, had two P53 variants, and shared one of those variants with this high-grade region. So that could be part of the story, but I'd say many cases where we're not seeing that, right? These are areas where we see P53 here, but not in the high-grade region. So that's certainly not the entire story. And I think it's worth going through a little bit more in depth just to kind of get a sense of how this is working. You can see just looking at the mutations that are common. And so for this patient, you'll see that MEN1 DACs are truncal, right? And they sort of are across, sorry, I'm showing you variant allele frequencies, and these are all the specimens. And you see that the variant allele frequencies are high across all. And so this is the truncal mutation. And then at some point, something happens where TSC2 has two variants, one here, and that seems to give rise to this low-grade region, and then this separate region here, and then this region sort of separately is divided by this P10. You also see this interesting thing I showed you before. DAX and ATRX are sometimes in the same sample, which obviously just means that there's like subclonal architecture within each of these as well. And so more to sort of be done about that. So just to summarize, transformation I think is common. Doesn't seem to be multiclonal. It seems to be this sort of very branched pattern with early diversity. That might explain some degree this, the surgical debulking might explain some degree this early emergence of the high grade. But I think there's some cool things going on there as well with the dormancy. And then P53 it's common and are transformed, but it's maybe driving transformation in a few instances, but certainly not the whole story. There's a few other things we're working on. Maybe just in interest of time, I'll just skip through all this and just thank you.
Okay, thanks so much. Can you hear me? Okay, so we have time for questions. Etay, would you like to come back? This is the difficult part. We're going to question you now.
Oh, it's done. Get my breakfast. Oh, sorry. Sorry, I didn't see you.
I was just wondering if there was any there were any copy number changes or anything like that you saw that preceded the transform that from your phylogenies you would assume might precede the transformation that might sort of prime.
Yeah, we're tracking
higher probability of that.
Right. Yeah, we're tracking the copy numbers as well. We haven't done. This triomics is a pretty nice algorithm for this, or sort of been used in a lot of cases where it sort of seems to do pretty well for these sort of phylogeny reconstructions. They're not as great ones for copy number aberrations, although there's some. But certainly, given these phylogenies, we can then look at the time and the copy number, and we're looking at that now. They're not that great copy number aberration sort of phylogeny inference algorithms.
Mauro?
Great talk. When you look at the increase of Ki67 over time, do you use only surgical specimens or also biopsies?
So it's both. I think the majority are surgical. but there's also some that are biopsies. And certainly there's heterogeneity in the biopsies. I don't know if that was your question, but that's certainly the case. Although truthfully, I mean, it would be nice to have the full surgical specimen always. And actually I didn't show this one, but there's another patient this is actually work with Chris Iacovos. I don't know if you know her, but she does this stuff in PDACs and has this whole program of autopsy, basically patients who sort of, agree that once they die, they sort of give their body for this project. And they basically, if you do the autopsy and sort of have full view of the whole, the whole final sort of look at the cancer. But to your question, I mean, you certainly are seeing a small sample, right, when you're doing that. But in the end of the day, you're just interested in the high grade. And so if you're seeing high grade transformation, then, again, you're seeing a small part of it, but at least you can see where it's mapping on the phylogeny.
Thank you.
Great work. I really appreciate what you're doing. Many of us have had the experience where patients get treated with an alkylating agent or PRRT and they show that takeoff. Actually, your curve that you showed a subset of patients that did that. Is there any signal that you can see in the primary of those who are most likely to do that?
Yeah, it's too small to say much. And I think I'm also a little worried that it's biased because it's like I'm looking at this one subset. And also when we biopsy it, or when we so there's the surgeries and then the biopsy, and the time of the biopsy is going to be typically after our treatment, which I'm an interventional radiologist, so like embolizations, because it's convenient and safe to do it at that point. I don't think there's much that I see. I thought maybe alkylating agent would be something. I certainly don't see anything related to the radiation. I kind of lumped it as PRRT or any other radiation, like a radioembolization or something, or even SBRT, some of them had. And with the alkylating agent, I don't see anything clear. I thought maybe there was some signal with, I combined embolization with sort of Sutent because it sort of, I thought might change the tumor microenvironment, although I saw a talk yesterday that made me think that the microenvironment is changing probably very differently for those two. So I thought maybe there was a signal there, but too early, I think, to tell. That's part of the reason why I want to get at those other 15 that we're adding. But the hypermutation thing is interesting. I just add, Diane Reedy, who's one of our colleagues, they publish this exceptional responder stuff, right? So there's a sub cohort there that gets these amazing responses and disappears. I don't know if your patients, it's amazing, right, for years. And certainly some of those do eventually go into high grade, but I don't see a relationship between like hypermutation and, or at least a direct relation between hypermutation and this transformation.
So we'll have time for one more question.
Just real quick. I wasn't quite sure at the beginning how you decided to study these. Is there a bias that they're being maybe referred to Sloan Kettering because their disease is progressing? Because I thought that number with transformation and the number that remains stable at grade 1 was not what is my experience.
Right, so you certainly may be right. I can tell you, first of all, there's a bias for me, right? I'm an interventional radiologist, so I get this reference patients from Diane. And so like those are patients that failed her treatment. So I thought maybe that was part of it. Although when I asked her and Nitya, like they're like, no, we're seeing this very commonly. We think it's very common. But you're right, maybe MSK is like, overall biased, and it certainly would be nice to sort of get, do this in another cohort and see. I think there's other part of this is just to also look at patients who are sort of stayed, and there's some in our cohort, right, that have stayed for a decade or more, low grade, and haven't changed. So, okay.
Great, thank you so much. So now I would like to welcome Dr. Allison Stewart. She's an associate professor at the University of Texas at the MD Anderson, and today she will talk about the chemoresistant persister cells in the relapsed high-grade NECs. Welcome.
Hi, thanks for the introduction, and I'm happy to have the opportunity to share some of our work with you today. So, similar to the previous talk, but instead of transformation, we're very interested in studying high-grade neuroendocrine carcinomas. So these are clinically aggressive and they arise most commonly in the lung, but there are rare instances where they arise in extrapulmonary locations. So not typically, these are either large cell or small cell in histology, And currently, there are a few evidence-based treatment paradigms for extrapulmonary high-grade NETs that beyond those that are extrapolated from small cell lung cancer. So these are just some of our top questions and the serious unmet needs for high-grade neuroendocrine carcinomas. So primarily, there is an absence of samples and models with which to study this disease. So we've taken a system that we originally set up for small cell lung cancer, and we've expanded it to include pulmonary large cell as well as extrapulmonary high-grade NETs. We identify patients and consent them to our existing lab blood and tissue collection protocols. And we isolate blood, CTCs, generate PDXs from the circulating tumor cells, as well as banking plasma, buffy coat, cryopreserving PBMCs, and collecting patient biopsies. Primarily, these are core needle biopsies, but occasionally surgical resections. Then we've really tried to address a number of molecular analyses based on what the sample types are. So just to give you an idea, over the past 2 years since we started collecting extrapulmonary high-grade NETs, we've collected samples from, or over 100 samples from more than 50 patients. We've also generated a good number of models from these patients, including 2 circulating tumor cell-derived PDXs from extrapulmonary high-grade NETs, which really suggests that these patients have a good number of circulating tumor cells similar to what we see in small cell lung cancer. This is single-cell RNA-seq from high-grade NET patients as well as PDXs. And you can see that there's a high expression of neuroendocrine genes. Oops, wrong button. High expression of neuroendocrine genes, as we would expect. We also see heterogeneous expression of some of the small cell lung cancer molecular subtype markers, including ASCL1 and NeuroD1. And we see expression of YAP1. So this does not define a molecular subtype in small cell lung cancer, but we do see expression in these other high-grade NET samples. And we've done quite a bit of work on YAP1. I talked about it a little bit last year, but in high-grade NET samples, YAP1 defines a subtype that makes up roughly 50% of both pulmonary, oh my goodness, I'm pushing the wrong button, sorry. Pulmonary high-grade NETs, as well as using some publicly available data sets for extrapulmonary high-grade NETs. And YAP1 defines either high or low subsets that are characterized by molecular features, including EMT and immune cell infiltration. And I think the most exciting thing is that there are therapeutic vulnerabilities that are related to the YAP status from these patients. And this really offers hope for personalized therapy going forward. Another question that we've really focused on is that there is a great need for increased cytotoxic therapy strategies. So these high-grade NET patients typically respond very well to platinum-based chemotherapy, and then they relapse. And at relapse, they don't respond to anything. Additionally, immune checkpoint blockade is rarely effective in these patients. And so we've been interested in exploiting the surfacome as an alternative way of targeting cancer in these patients using either antibody-drug conjugates, CAR-Ts, or bispecific antibodies that recognize cell surface proteins on the cancer cell as well as T cells. And we know, looking at a cohort of patients that were collected at MD Anderson that we've profiled, A number of the common cell surface targets that have been explored in other solid tumors, including HER2, TROP2, are expressed in these patients, and they do seem to be distinguished by the YAP status of the patients. So to take this farther, these are high-grade neuroendocrine cancer cells that are defined as being either YAP low or YAP high. And we can see differential expression of some of these surface targets, including AXL, DLL3, and CD56. And when we test these cell lines using CAR-Ts, even though there is not a significant difference in DLL3 or CD56 in the YAP low versus high cell lines, we do see significant cytotoxicity in the YAP low cell lines. And conversely, AXL is, the AXL CAR-T is more effective in YAP high cell lines. And so this really suggests that YAP status predicts benefit with CAR-Ts as long as you're targeting the correct population. And when we look at our single-cell RNA-seq data in the high-grade NET patient biopsies, I think this kind of strengthens the finding because the red cells are expressing AXL in both of these plots, and the green cells are NCAM or CD56, and the blue cells are DLL3. And you can see that they're mutually exclusive expression patterns. And then finally, more related to the title, we have a poor understanding of this resistance in these patients. So as I mentioned, the tumors are very recalcitrant, and that occurs following the initial response. So this is suggestive of persister cell populations. And we've studied this probably more in small cell lung cancer. And as I mentioned previously, YAP1 is not expressed in, or is not a subtype defining transcription factor in small cell lung cancer. So it's not expressed in treatment naive disease, but at recurrence, then YAP1 is expressed. And in small cell lung cancer, YAP1 is expressed specifically in these highly plastic populations. So the RNA velocity is greater in YAP1 positive cells in small cell lung cancer. And to take it back to the surfacome targets, we see that some of these surfacome genes are co-expressed in YAP1 positive populations. So specifically B7H3, TROP2, and HER2 are expressed in the same populations that YAP1 is expressed in. And sorry, YAP1 is the red cells in this case, and the blue cells are the other targets. Whereas some of these other cell surface targets are not co-expressed with YAP1. So this suggests, at least in small cell lung cancer, that there are some actionable cell surface proteomes within the resistant or plastic cell populations. And so we've been trying to apply this to some of our other high-grade NET models. And as I mentioned, we hypothesized that drug-tolerant persister cells are occurring in this disease. So when the patients respond well to platinum chemotherapy, then the number of cells, so most of the cells die off, but there is a population that are sticking around, and then these are able to repopulate the tumor. Drug-tolerant persister cells typically are described to evade death by entering a reversible, slow proliferative state. And this allows these residual cells to survive. And so to study this, we've been using this really nice construct described by Oren et al. 2 years ago. It's simply a DOX-inducible mCherry system. So cells express mCherry, and as the cells proliferate, the mCherry is diluted out, which allows us to identify high cycling and low cycling populations. And so we've done a simple experiment with a DOX pulse chase During the chase period, as the cells are proliferating, we're hitting them with therapeutic pressure, either DMSO or high levels of cisplatin and etoposide at an IC70. And after prolonged exposure, we've sorted the cells for the cells that have high M-cherry expression versus low M-cherry expression. And as we would predict, the cells with high M-cherry have an expansion of the G1 phase, which suggests that they are non-cycling persister cells. And when we look at the cell surface markers in these cells following the therapeutic pressure, or in the DMSO cells, there's no expression of AXL or TROP2, but with therapeutic pressure, we see an increase in cell surface expression of both of these, and both of them are increased in the non-cycling or high M-cherry cell population, suggesting that there are some cell surface targets enriched in these persister cell populations, which gives us hope of being able to target them. So in summary, we've curated a cohort of patient-derived samples and models. We find that these high-grade NETs exhibit distinct biology based on YAP status, and that CAR-T therapies are effective when delivered to the appropriate cohort of samples. Chemoresistant persister cells exhibit a unique actionable cell surface proteome. And some of our future efforts are to do mass spec analysis of some of our novel high-grade NET models to identify cell surface targets that may be specific for extrapulmonary high-grade NETs. Additionally, we're in the process of assessing BiTEs, CARs, ADCs, to target these drug-tolerant persister cell populations as a way to mitigate resistance. And excitingly, there's been an expansion of clinical trial efforts in the extrapulmonary high-grade NET space. So we currently have a number of clinical trials that are for small cell patients, but that include an arm that allows for extrapulmonary high-grade patients. And this is just a few of these. So thank you very much, and I'd be happy to answer questions.
Thank you. Great presentation. We have time for some questions. Yes.
Yeah. Thank you, Allison. Nice overview. My name is Jules Derks. I'm from Maastricht, Netherlands. I've been working on LCNEC, pulmonary LCNEC, for a while, and the research that you're using, all the cell lines, state that these are LCNEC. How sure are you that these lines actually represent pulmonary LCNEC? Because I have severe doubts on this. We have looked into these cell lines also, and I've not seen correlations with their morphology. So really seeing pulmonary, so the neuroendocrine morphology, once you inject these models into mice, and are these adenocarcinomas with a bit of neuroendocrine differentiation? Can you explain on this?
So we received the cell lines from UT Southwest, and they're the ones that made the cell lines. So we're fairly confident based on that to start with, but I disagree. We do see a difference in phenotype between, like, our YAP high cell lines are more attached, and the YAP low cell lines tend to be more small cell-like, and they float. So we definitely see a difference in the behavior of the cells.
I agree. We're doing some similar analysis, so I agree that there is this big difference for YAP positive and negative cases, but I'm just not sure if we can really state that these cell lines are LCNEC, because there has not been a morphological comparison of these cell lines, at least to my knowledge of current literature.
Okay.
But it's relevant, I think, to do.
Okay.
Maybe I will ask a question. Maybe I've missed it, but so you have conversion from YAP low to YAP high. Do you see that in your samples?
So we see that in small, well, I don't know if I would say YAP low to YAP high. In small cell lung cancer, they basically don't express YAP1, but then in resistance, we're seeing an emergence of cell populations that express YAP1, and these are highly plastic and express senescence-associated secretory protein genes and stem cell markers.
But in the extrapulmonary, do you see the same?
No, actually what we see in extrapulmonary and in pulmonary large cell is that they start out even treatment naive with high expression of YAP1, but not all of the patients. So it's typically like 50-50.
Okay, great. Thank you so much.
Thanks.
Great. So we're going to move forward to the next speaker, Jeanna Q. Okay. So the Jeanna is an MD PhD student in the William Hwang lab at Harvard, and she will be discussing molecular landscape of pancreatic neuroendocrine tumors at single cell resolution.
Hi, everybody. My name is Jeanna Chu, and I'm a second-year graduate student in the Harvard-MIT MD-PhD program. And today I'll be sharing our work on the molecular landscape of pancreatic neuroendocrine tumors at single-cell resolution. So pancreatic neuroendocrine tumors, or PNETs, are caused by the abnormal growth of endocrine cells in the pancreas. And PNETs are a very diverse group of tumors. They're both phenotypically diverse in that about 10% are functional, producing hormones and causing a vast array of diverse symptoms, while 90% are non-functional and not producing any hormones. And these non-functional tumors can often grow for a long time asymptomatically and therefore are caught often at later stages in disease. In addition, PNETs are also very clinically diverse. While most PNETs are considered to be indolent and slow-growing, some are quite aggressive and can metastasize quickly, as we just saw. And furthermore, even though features like mitotic index or proliferative like Ki67 and ALT are associated with prognosis, we still do not completely understand the molecular features relating to prognosis and metastasis in PNETs. So some of this clinical heterogeneity may be driven by molecular heterogeneity within PNETs. So previous groups have tried to characterize different subtypes of PNETs. There is the alpha cell-like subtype driven by the transcription factor ARCs and associated with mutations in MEN1, DAX, and ATRX. They've also identified a PDX1 high subtype driven by the transcription factor PDX1 and resembling more beta cells, as well as a proliferative subtype associated with poor prognosis and a stromal and mesenchymal cell type. However, one limitation of these previous studies is that many of them were done in bulk. So as a result, this results in the mixing of all the different cell types within the tumor sample, and we can't understand the specific features of malignant neuroendocrine cells when using these methods. So overall, there really is an urgent need to understand the molecular heterogeneity in PNETs and also how this is related to metastatic potential. And then in our labs, we have established a growing biobank of mostly non-functional PNET surgical specimens. So to this end, we've applied single nucleus RNA sequencing to a subset of our biobank with the goal of comprehensively defining the transcriptomic landscape of PNET at single cell resolution, and also to comprehensively understand the malignant cell states in PNET. So we applied single nucleus RNA sequencing to about 20 PNETs in our cohort, and after quality control, we ended up with a cohort of 15 primary resected, untreated PNET specimens. This is mostly non-functional PNETs and includes one glucagonoma, and they encompass a really diverse range of grades and stages. So we recovered 91,739 high-quality single nucleus specimen profiles from these 15 specimens. And this is a UMAP representation, which is a two-dimensional representation of the transcriptional profiles with each dot representing a cell. And here it's colored by patient ID. We then wanted to look into what are the different cell types And so first we identify the different compartments by looking at the expression of canonical marker genes. So in teal, you see the malignant compartment. In purple is the stromal compartment. In green is the immune, and in red is the epithelial compartment. Now we wanted to take a closer look within each of these compartments to identify the different cell types. So within the stromal compartment, using established literature markers, we identified 5 different cell types, including endothelial lymphatic and vascular cells, fibroblasts, and pericytes and vascular smooth muscle cells as well. And then in the epithelial compartment, we identified acinar and ductal cells, as well as a large population of acinar idling cells, which is a subset of acinar cells. And then we also identified various lymphoid cells. So the top row is the myeloid compartment with mast cells, neutrophils, monocytes, and macrophages. And the bottom row is various different lymphoid cells that we identified. So now that we've characterized the different cell types in the non-malignant compartments, we wanted to take a closer look at the malignant compartment. So as I mentioned, we identified the malignant compartment through the expression of different marker genes. And then we also used infer-CMV to infer copy number variants from the single nucleus profiles, as we would expect that malignant cells would have a much higher copy number burden compared to the non-malignant controls. So here the top row is the reference of all the non-neuroendocrine cells, and the bottom is an example of the copy number profile of one of our specimens. And as you can see there, with the red representing amplification and blue representing deletion, there is much higher copy number burden in the malignant cells compared to the non-malignant cells. And the most common copy number alterations that we see in our cohort are 11q deletion, which is in the MEN1 locus, 10q deletion, which contains P10 and MGMT, as well as 7q amplification, which is the HGF locus. So now that we've identified the malignant compartment, we wanted to understand what are the different cell states of malignant neuroendocrine cells. So to do this, we use something called consensus non-negative matrix factorization. So essentially what this does is it is an unsupervised clustering method to infer gene expression programs. So it takes the matrix from single-cell RNA sequencing of cells and genes, and then it decomposes that into 2 matrices. One contains gene expression programs and just defines each program by the level of expression of each gene. And the other matrix is the level of activity of each of these gene expression programs in each of the single cells. And to make sure that we're identifying true biology and not noise, we do this many times. In fact, we do this 200 times. And then we find the consensus solution that's shared across all of these replicates. One of the challenges in performing CNMF is deciding K, or the number of gene expression programs. So in our case, we focused on gene expression programs that were shared across multiple patients to inform general biology about PNETs rather than patient-specific biology. So overall, we identified 7 gene expression programs with CNMF, which are seen on the bottom. They're the columns in this heat map. And first, we wanted to compare it to known transcriptional signatures. So the top 4 rows are the bulk RNA PNET signatures that I described earlier in this talk, and the bottom are single-cell transcriptional signatures of normal neuroendocrine subtypes. And we identified one program that had significant overlap with the beta cell signature. And this contained a lot of genes related to GPCR signaling or insulin secretion. The purple in the UMAP represents the level of activity of this program in each cell. The program that had the highest overlap with the alpha cell-like signature contained a lot of genes related to inflammation and cytokine signaling, including CXCL9. Then we identified 3 programs that we typically see in most cancer cells. So 2 of these had overlap with the proliferative signature. One was a ribosomal gene expression program containing a lot of different ribosomal genes. Another was a cell cycling signature that contained a lot of genes related to the cell cycle, M phase, and the mitotic spindle. And then the third program that we typically see in most cancer cells are mitochondrial genes. So genes related to tRNA and RNA processing in the mitochondria, as well as the TCA cycle. Now, what was interesting is that then we were left with two programs that did not have overlap with previously identified signatures. And so these were representing novel gene expression programs in PNET. So the first was this neuronal and synaptic signaling program, which contained a lot of genes related to phosphatases at the synapse or scaffolding proteins at the synapse, as well as glutamate receptor molecules and proteins related to axon guidance, including netrin G1, which is a family in the netrin proteins that were described yesterday, as well as UNC5D, which was also described yesterday. And then the last novel program we identified contained a lot of cytoskeletal genes, so including a lot of actin-binding genes like myosins, tropomyosins. But another significant term for this program was VEGF-A signaling, which is interesting considering that a lot of the current therapies are targeting this pathway. So overall, we're presenting a refined taxonomy of malignant cell states in PNET, with 2 representing previously identified signatures, alpha and beta cell-like, 3 that are typical that we see of most malignant cells, and 2 novel programs. The next step that we're currently doing is identifying if any of these cell states have clinical significance and specific therapeutic vulnerabilities that we can target. So we're doing this with validation with a cohort of bulk RNA sequencing, PNET specimens, and also of other neuroendocrine neoplasms. And we also want to fully characterize the other compartments of the PNET microenvironment and investigate interactions between them. And then finally, it would be really interesting to understand the interaction between oncogenic mutations and cell state with this refined taxonomy of cell state. Whoops, sorry. So with that, thank you very much. I would like to thank my PIs, Will Hwang at Mass General and Martin Hemberg at Brigham and Women's Hospital. And from Will's lab, I'd like to thank Jennifer and Jimmy, who did the sequencing, Karina and Stephen, who are former members of the lab who started the project with the analysis. And from Martin's lab, I'd really like to thank Jaewon Cho, who's a postdoctoral fellow who's really been mentoring me through the analysis. And thank you to NETRF for the opportunity to present and also for funding our project. So thank you.
We have time for questions.
Yeah, thank you. Thanks, that's really wonderful. Just 2 things. First, your thoughts on, you guys use single nucleus. Is that from a technical thing or more biological? Do you feel like you miss some stuff if you're doing single cell, like, or the whole cell?
So doing single cell RNA sequencing in the pancreas in general has been really challenging because it produces a lot of digestive enzymes. So our lab had previously refined a protocol for doing single nucleus RNA sequencing in pancreatic ductal adenocarcinoma specimens, and we applied that same protocol to the primary PNET specimens.
One other quick thing, the K, it's a common problem for any clustering. There's some very good automated ways to sort of let the data tell you how much clusters there are, like information bottleneck or something like that. Those are some other things to kind of try for clustering.
Yeah, so what we did is we looked at the stability of the solution and also the error of the solution and tried to minimize error, increase stability. And then of those programs, we focus on the ones that were shared across multiple patients and not just clustered in one patient.
And what was the K ultimately then for that? Or?
So the K ultimately was 25, and we had 15 specimens, so there were 15 programs that were just clustered around one patient, and then we performed further downsampling to look for the consensus of the programs that were stable, and that led us to those 7 programs.
Next. Great talk, congratulations. So just a curiosity, I know you said you are already working on studying a little more the immune microenvironment. I was wondering if you have preliminary data on immune checkpoint, on T cells, but especially on the tumor cells or the myeloid cells, because there are a few recent articles that are showing that there are other immune checkpoints like TIM3 or VISTA on these cells. So I was just wondering if you can give me your feedback on your cohort.
Yeah, that's a great question. We haven't fully started characterizing the immune microenvironment or the checkpoint inhibitors expressed in our cohort, so I can't really tell you right now, but there has been some really exciting work in single cell of neuroendocrine tumors showing some the expression of different immune checkpoint inhibitors. So,
Looking forward to know your
Thank you.
experience.
Thank you.
Last question?
That was really exciting, thanks. So I was really struck by the inter-tumor heterogeneity between your different patients. And I'm wondering if you could just speak briefly to the intra-tumor heterogeneity you might be seeing in the transcriptional states.
Yeah, that's a great question. So like I mentioned, we haven't specifically looked at using, for example, something similar to CNMF within each of the patients, but that's actually something that we're considering doing next. So I can't really speak to that right now, but that's a really interesting question. And for the first purposes, we were trying to focus on general programs that are shared across neuroendocrine PNETs that could explain general biology, but that's a really great question to look into in the future.
All right, thank you, Jeanna.
Thank you.
Okay. So our last speaker in this session will be Chang from Rutgers. He will be discussing single-cell transcriptomics, identified IGF2 and VEGF as growth factor in pancreatic neuroendocrine tumors.
Thank you. I would like to thank the organizers for putting together this great symposium and for giving me the opportunity to share with you some exciting research we have that are unpublished. So this morning, Etay and Jeanna gave you a very nice introduction to PNETs. So as Etay showed, the most commonly mutated genes in PNETs are MEN1, DEX, and ATRX. In published work from 2018, we showed that when you take RNA sequencing from both tumors of PNETs and you do an unbiased clustering to identify, what you identify is two very distinct subgroups. And what surprised us was that all the tumors with mutations in ATRX, DEX, or MEN1 lies within one subgroup. And so that's, furthermore, we showed that that subgroup had a gene signature that was like was alpha cell-like. The other subgroup had more of the PDX1 like signature. But what we still don't know in this case is what is driving the tumor growth. And moreover, if we want to understand what is the gene expression dysregulation in these tumors, knowing the proper cell type to compare it to is crucial. And we know that in many cases in these tumors, it's not going to be the beta cell. So that led us to the idea of actually using single cell approaches to study these questions. And to do that, we teamed up with Stephen Labuti's lab from Rutgers Cancer Institute to collect fresh tumors, PNETs, from surgical resections and perform single cell RNA sequencing. So today, I'd like to share with you some results we have from 7 such samples. And if you look at these samples, you'll see that 5 of these are well-differentiated non-functional PNETs. 2 of them are insulinomas, and they're all mostly low-grade tumors. We perform exome sequencing on these tumors. And what we identify is that some of these tumors have mutations in ATRX and MEN1. We were able to successfully develop a protocol to get good quality RNA, single cell RNA sequencing from these samples. And what I'm showing you here is the cellular population in these PNETs that were identified. So if you notice the top row of 3 and the next row of 2, these are the non-functional well-differentiated PNETs. If you see, we identify that 3 of these PNETs have tumor cells that look like the alpha cell-like, alpha cells. And 2 other tumors, one of them has cells that look like gamma cells, And there's another tumor that has some mixed cell populations with some beta cells and some gamma cells. In contrast, the insulinomas clearly have they have primarily beta cells. Now, the way we identify the cell type is because with these some of these non-functional tumors, they don't express any of these typical neuroendocrine hormones. So to find the cell type, we essentially compare the global transcriptome to see what's the closest match, whether it's an alpha cell, beta cell, gamma cell, delta cell, or epsilon cell. And the comparison comes from published data of these normal neuroendocrine cells from a Regeneron group that we are using. And so the data that we're using on the bottom right panel is the alpha, beta, gamma cells from this published paper in 2018, where they did single cell sequencing using the same platform, 10x platform that we use for our samples. So the first thing we did is in each one of us of our PNETs, we isolate from the data the tumor cells. And if it is an alpha-like cell, we're going to compare it to normal alpha cells to find what's the dysregulated gene expression. So here's an example of one such tumor. In the top left panel, what we're showing here is only the tumor cells from this PNET that has an alpha-like signature. And as you can see, there is intratumoral heterogeneity, right? So there's, from this clustering, we identify perhaps 3 different subclones of this PNET. And what's interesting is that in one of these subclones, there's differential expression of a set of genes we show in the heat map, but one of the genes that caught my eye is IGF2, right, which is insulin-like growth factor 2. And furthermore, what we can do is you can do cell-cell communication using known pairs of receptors, ligands and receptors to identify whether these cells are communicating to each other within the tumor microenvironment. And if we look at the growth factors, there are 2 that really popped up. One is there's a growth factor of IGF2 that is being secreted by the tumor cells. And the receptors for that are many of the other cell types in the tumor microenvironment, but also the tumor cells themselves. So this is indicative of some autocrine signaling as well that's going on. The other growth factor that we see is the VEGF A and B factors. And these are secreted by the tumor cells, and the receiving cells are the endothelial cells. Now, this is the analysis that I described where we take the tumor cells that in this case looks like alpha-like, and we compare it to normal alpha cells. And so as you can see, we looked at these pathway enrichment analysis, and some of the top hits, I just want to highlight is there is signature for hypoxia. That's one of the top hits in these tumor cells. And also from the Reactome database, we see that another top hit there is regulation of insulin-like growth factors, right? Which is consistent with what we have found from the cell-cell communication. Just to highlight another, this is now a study where we take, within the intratumor heterogeneity, we can take the cells that had higher expression of IGF2 compared to the tumor cells that have lower IGF2 expression. And here what we see is that some of the hallmarks of the genes with high IGF2 is that there's MYC target genes, for example, right? And a lot of genes are involved with translation, with protein translation. Now, going forward, this is just to give you another example. This is a tumor that instead of being alpha-like is actually a gamma-like cell. Now, mind you that from the transcriptional picture, among the neuroendocrine cell types, gamma cells are very similar to alpha cells. So in this case, again, very similar picture to what we find before. There is intratumoral heterogeneity. Again, in one of the subgroups, IGF2 is a little bit more higher expressed than other subclones in this tumor. And the same two growth factors again pop up where you have IGF2 and VEGF signaling. Likewise, when we did the comparison between the tumor cells that are gamma-like to normal gamma cells, we're seeing again the signature for hypoxia in one of the cancer hallmarks, and we see again, and from the reactome database, the regulation of insulin-like growth factors. And then when we do the comparison between the IGF2 high tumor cells to the IGF2 low tumor cells, again we see, in this case we see more oxidative phosphorylation, hypoxia. Mixed signaling is also found there as well, and eukaryotic translations category in the reactome. Now, this is a summary of, again, of the 7 tumors that I am presenting today, and among these 7 tumors, 5 of those are the well-differentiated PNETs, and 2 of them are insulomas. In the 5 PNETs, well-differentiated PNETs, 3 of them, as you can see, express high levels of IGF2. In contrast, when you look at normal alpha, beta, gamma cells, they do not express high levels of IGF2. So this is tumor specific. Similarly for the VEGF-A and VEGF-B. For the endothelial compartment, we can show that there's also a higher expression in the tumors, in the PNET tumors, for the receptors of the VEGF signaling. And these are expressed higher than the endothelial cells that you find in normal tissue. Now, what I presented was about 7 samples, but what I can show you is that if we take a larger group of PNETs, and this data comes from the data we have previously published in 2018, where we did bulk RNA sequencing of a large number of non-functional PNETs. We find that if you look at the expression of IGF2 in these tumors, about 1/3 of these tumors have very low IGF2, almost no IGF2 detected. About 2/3 of them actually have IGF2, with some with very high levels of IGF2. This is not specific to either the subgroup that has, the alpha-like signature, or the other half that is well-type, the well-type for, ATRX, DAX, and MEN1. So it's irrespective of that mutational pattern. The second thing I want to point out is that you may notice that the IGF2 is located in the genome very close to the insulin gene. In fact, the insulin gene is upstream of the IGF2 gene. So people have claimed that it could be a result of transcriptional read-through. If you have high levels of insulin, the transcription could just, as a byproduct, produce high levels of IGF2. We showed that in this case, it's not the case. If you plot on a 2-dimensional plot for each sample, the expression of insulin and the expression of IGF2, there are samples with very high insulin and no expression of IGF2, and there are samples with very high IGF2 and no expression of insulin. So it's not coming from a transcriptional read-through. Right? So lastly, I just want to come up with a potential model that can explain what might be going on here, which again, this is a little more speculative now, and is that the most common copy number alteration in these PNETs is a loss of chromosome 11. And part of the reason for that is that MEN1 is found on chromosome 11. So MEN1, we know, it's an early mutation. It's a classical tumor suppressor. There's usually often a mutation in MEN1, and then what happens is that you lose the wild-type copy, usually by loss of the chromosome. Well, it turns out that IGF2 is also found on chromosome 11. But IGF2, moreover, is known to be a sex-imprinted gene, so it's only expressed in the paternal allele and not the maternal allele, and it's thought to be expressed primarily in the fetal stages of life and not in the adult tissue. So what could be happening is the early mutation of MEN1 with a loss of chromosome 11, if you end up losing the paternal copy of chromosome 11, well, you lost the copy of IGF2 that's being expressed, and that explains the tumors that have no expression of IGF2. in the tumors where you lose the maternal copy and you retain the paternal copy, then you could have further tumor evolution that may then upregulate the expression of IGF2 for growth signaling. So that's sort of a working model that we have. So lastly, I want to acknowledge all the people that have worked on this project from my group, especially Fu Qian, and from Dr. Stephen Labui's group, Ziqi and Yuan have helped us a lot with getting the fresh human samples. And of course, I'm very grateful to NETRF for funding this research. Thank you.
Beautiful talk, Chang. So I was wondering whether you ever correlate your IGF2 and VEGF expression levels with tumor grades or metastases.
Yeah, so unfortunately we have, that's something that we can do. In our samples, there are 7 samples, they're all very low grade, but you're right. What I'd like to follow through is with a different data set where there is more variable grade and see whether that's, yeah. Thank you.
Thank you for a great talk. I just had a quick question. Similar to that, is there any data on the pancreatic neuroendocrine tumor samples that were selected and whether they received somatostatin analog therapy? Because if they were on SSAs leading up to the time of resection, potentially that could have an impact on sort of the IGF pathways.
Yeah. So again, in our data set, again, these are all very low grade. So these are all previously untreated patients.
Okay.
Right. Thank you.
So I think maybe if we can get all the speakers up here now, and then we can open the floor for any other questions, and maybe we can have some discussion. Yeah.
I think Scott can start.
Yeah, is this working? That was a great session, and I wanted to just raise this conversation a little bit around heterogeneity in this tumor. Pancreatic endocrine cells are extroverts. I mean, they develop in this important community with a lot of different cell types around them. The exocrine endocrine communication is extremely important in the pancreas. We see, for example, when you patients that lose exocrine function, their endocrine cells are eventually going to degenerate, right? So there's lots of signaling, and that is now becoming more and more apparent. There's NIDDK is all over this lately. But in this tumor, I think it's important to understand that. So I love the fact that you guys are really starting to take a look at tumor heterogeneity. It would be very interesting to see, I think, what happens. First of all, to be able to quantify intra-tumor heterogeneity transcriptionally, quantifying this, and also looking at what happens to it for after treatment in the metastatic lesion versus the primary lesion, and then just generally keeping our mind open to what is it doing for the tumor. We're always assuming, for example, that we should not assume, for example, that the cells that are sensitive that we're able to say treat and remove initially, that they're not necessarily doing something to control the more aggressive cells that are in the tumor. There's other instances recently where in other cancers we're seeing that if you remove the sensitive cells, you promote the emergence of the resistance clones, right, from this. So anyway, I just wanted to raise this conversation. I love that you guys are starting to really get into looking at tumor heterogeneity, but I'd love to hear anybody that had any thoughts on this. I don't think your thing is on. Yeah.
No, I just saying I totally agree with everything you're saying. I think that's direction we're all heading towards. And I definitely agree with the idea that removing some of the sort of, let's say, easier to treat areas may be sort of disinhibiting the more aggressive areas too, and all that. I totally, yeah. I don't have any insight on it. I'd say I agree.
Okay, thanks. Thank you.
So I guess I'll make a comment. I would like to see what the speakers think about this. So in 2021, we published a paper actually in which we show that during the evolution of pancreatic neuroendocrine tumors, you have now these functional insulinomas that then they switch to the non-functional through DD differentiation. And then during the switch, actually, they keep the same Ki67, the same proliferation. And then you have a second step when they become hyperproliferative. But what was interesting is that this switch between actually the functional to non-functional and the differentiation was not driven by any mutations. It was driven by microRNAs. So I would like to first think, what do you think should we start looking more into epigenetics, maybe methylation, microRNAs, whether they are driving both the persistent clones and also these high-grade non-functional tumors. And then one thing I think nobody looked into it is maybe start looking maybe at the progenitor-like signatures, and maybe that's what will explain a lot of the heterogeneity instead of looking at the classical alpha cells, beta cells, and gamma cells. So I would like to see what you think.
Yeah, no, I think these are great comments. So for us, like when we were trying to identify one of the reasons behind this was trying to identify what's the cell of origin for some of these tumors. And it's, I compare it to essentially a group of cells that are fully differentiated, right? From adult tissue, alpha, beta, gamma, right? And, it would be great to get more data on what some of these earlier, progenitor cell transcriptome states would be, so that we can get a perhaps even closer match, a better understanding. So I think generating that kind of data may be will be very important.
Yeah, I completely agree. Another thing that it's really hard also to analyze, like, for example, non-coding genes or non-coding RNAs, microRNAs. I think all of these might be playing a role, but the analyses that I've been doing are mostly focused on protein coding genes, but I certainly see a lot of non-coding RNAs. And so that's something I think would be really interesting to look into.
Yeah, and also in addition to Scott's comment previously, I think investigating and comparing between primary tumors and metastases is so important, especially for the tumor microenvironment. The blood vessels and other the tumor microenvironment components can be very different depending on the tissues, like a lymph node or livers, lungs, and primary tumor in pancreas. So I know it's technically hard to get the paired primary and metastatic tissue for analysis, but I think that's the way that we should go future. And another thing is that for the Etay, I have a question, because I think it would be very important to identify the signature in tumors where we can predict, the potential of disease progression and also metastasis, and even more like a metastatic tropism, like a lymph node versus a liver metastasis. Well, what do you think? Is it?
Yeah, no, I totally agree. We used whole exome on our side just more to be able to, try to tease out the relationships. I don't think that that's necessarily driving anything. I think the tropism and the relationship with sort of the microenvironment is sort of, that's my sense is where a lot of this is happening. And so absolutely breaking things down by sort of where the tumor is. And I think each of these methods are maybe like a way to sort of get at what we all kind of want in the end, but.
Yeah.
Allison, I had a question. So how about YAP1 expressing tumors? What do we do with those?
Yeah, sorry. So we've found in our YAP high subset that CDK4/6 inhibitors seem to work pretty well, and but I guess our interest right now is really using these surface targets. So AXL TROP2 ADCs is kind of the way that we're going after this. We like the idea of ADCs because some of those are approved, but we are currently, I guess, concerned because most of the payloads for ADCs are targeting highly proliferative cells, and the drug-tolerant persister cells are not highly proliferating. And so I think right now, I think like the BiTEs and some of those are really going to be the most translational options for patients. But yeah, those YAP cells seem to be bad news. So.
And are you able to turn off YAP in a YAP-expressing cell, and then what happens to the surface?
Yeah, that's a great question. So we've tried a number of YAP inhibitor, like verteporfin and some of these, and they don't seem to function well in the high-grade NEC samples that we've tested them in. They work just as well if there is no YAP around. So finding a way to turn off YAP1 or target those cells, is really important, I think.
And Etay, so you can't tell me a grade 3 has this mutational profile. That's bad, right?
You mean like, is there a mutation profile that defines
or
grade
what
3?
they're acquiring, you were saying.
I don't know one, no.
And then you just said you're thinking about the tumor microenvironment as a driver of sorts.
I mean, I think that's maybe playing a role, yeah.
Yeah.
I could say that I think the microenvironment in high grade and low grade is different. What we've seen, we haven't done single cell, we've done bulk. We'd like to do the single cell, but at least in bulk, if you tease out the microenvironment from that, which is, let's say, problematic, it's not great, but it looks like there's some differences.
Is there a difference in the epigenome between the grade 1s and the grade 3s?
Working on it.
Okay.
Good questions.
So maybe I will answer a couple of your questions from data we have both from the mouse model, but also some of the human samples, is that there is difference in the microenvironment between the G1 functional and the higher grade non-functional. So definitely the question is what is again this and the right, is what drives first this switch? Is like the cells, they switch to high grade and then they extract the microenvironment, or is it something that moves in these tumors and then causes the switch? In glioblastoma, they show that M2 macrophages are causing the switch from one subtype to another.
Oh, I know.
And macrophages are different between the low and high grade. Right. So that's maybe something that somebody can explore.
Hi, so we've learned a little bit about grade changes within NETs, neuroendocrine tumors. Can you talk a little bit about what we know or what we don't know about the potential transformation from a NET to a neuroendocrine carcinoma?
I mean, so I'd say I really just leave the decision about poorly versus well to the pathologist, because let's say in the clinic, I don't think that I see and that medical oncologists don't believe that these are ever transforming truly to poorly differentiated, and asking the pathologists, they say, no, that's still well differentiated. Although I say that with a little bit of a I don't know, the line gets a little blurry. Laura's original paper, it's clear genetic, distinction. That's not totally true, right? And so I guess I don't know. In the end, I think we'd have to ask a pathologist, they have their categories, but it gets a little murky. So I don't know. I don't think so. At least officially, no. I don't know if it's maybe possible that it still is.
Okay, thank you.
I guess again to comment on this, so I asked my pathologist to look at the samples the past, I think, 20 years. And they said they had only 2 cases where you have both well and poorly differentiated in the same sample. And it seems actually it was 2 different clones. So it's
cool.
But yeah. Any other questions, comments? Okay, everybody wants to go. Oh, yeah, go ahead.
So do you think IGF2 should be targeted?
I think that's a potential thing that we would like to look into.
Uh-huh. So, I mean,
They're all IGF2 inhibitors.
yeah, I mean, they haven't done so great, but maybe they were picking the wrong tumor.
That's right.
But that's an interesting observation. It's a straightforward explanation of sorts. So, yeah, okay.
I just wanted to follow up on the IGF2 question. For MEN1, we don't see a parent of origin effect. So in your kind of model, why would that be?
So, sorry, what is it?
So for MEN1, there's no parent of origin effect in terms of the phenotype. So if IGF1 is one of the main drivers, would you expect there to be one?
No, I mean, I think the parent, you're saying the parent of origin, right?
Yeah.
Yeah. So, so IGF2, as I said, right, is a sex imprinted gene.
So
we don't find a mutation in IGF2. We don't quite understand. I mean, a priori, right?
Sorry, I meant IGF2.
Okay, well, you mean IGF2, right?
Yeah. So 11, chromosome 11, SDHD is on the same chromosome, and you see a parent of origin effect, right? Because of the imprinting region.
That's right. That's right.
So if MEN1 is synthetic to that, why wouldn't you see a parent, or what's your explanation of why you don't see a parent of origin effect?
Oh, the reason is, I mean, so it's the paternal allele that's usually normally expressed, right? So, I mean, what happens, right, in the tumor is that it's a somatic mutation usually that is, taking out randomly one of the, even parental copies of chromosome 11. So, I don't see how that would lead to a
I can draw it out for you.
Yeah.
But if the tumor suppressor, the loss of heterozygosity is always on the other chromosome, so it depends upon.
Well, I think the loss of heterozygosity, I think, is just driven by which copy of the chromosome the MEN1 mutation occurs in. That's what I think.
Right.
So the MEN1 can occur, randomly either in the paternal or the maternal copy. And in that case, what you end up driving is the loss of the other parental copy. And, it may or may not be, the copy that you lose may or may not be the paternal copy. So I guess what I was trying to explain is that if you happen to lose the paternal copy, then you don't have a IGF2 that can be expressed. And in that case, we see tumors that have absolutely zero expression of IGF2. So I would say that in those tumors, right, where you have an initial, an early truncal mutation and MEN1 and lose the paternal copy, they need to find a different method to drive their growth that's independent of IGF2. It's only in the tumors where you happen to get a mutation in MEN1, let's say in the paternal copy, you lose the maternal copy of the chromosome 11. Now, the one copy of IGF2 you have left is the paternal copy, which is the one that normally is expressed, right? And in that case, there may be some additional kind of tumor, alterations that happen that can, somehow drive up, further increase the expression of the IGF2, perhaps. I mean, I'm not sure. Am I making sense to you? We can talk some more.
Maybe you can continue our coffee. So please join me to thank all the speakers for this amazing session.